Hello. Today we're going to go over Chapter 15. This is Lecture 14 regarding antimicrobial drugs. Now, as an introduction to antimicrobial drugs, there's a variety of terms we need to understand, some history to understand how much this has impacted modern medicine. When I talk about antimicrobial drugs, these are therapeutic compounds that kill microbes or inhibit their growth. Antibacterial drugs treat bacterial infections. Antiviral drugs treat viral infections. Although this is going to seem sort of repetitious, there is a point here. And that is, it leads us to almost the part of the chapter at the end, which is how come if I have a bacterial infection, I get my antibacterial drugs, but if I have a viral infection and I've got the flu, why would I want to ask for antibacterial drugs, antibiotics? Because they don't work on viruses, including the flu. Antifungal drugs treat fungal infections, and antiparasitic drugs treat either protozoal or helminthic infections. Now, in history, one has to understand a little bit more about Fleming than just the fact that, hey, he looked down and he said, wow, there's this plate here that has a mold on it, and the bacteria are not growing into it. They're being pushed aside. They're not thriving right nearby where the mold is. Fleming was a doctor who served in World War II as a physician. And you have to understand that when there were limbs that were basically injured by shrapnel, bullets, etc., a lot of times those limbs, legs and arms, yes, would get infected. And the only way to treat them was to cut them off. Now, afterwards, Fleming understood this very, very acutely, very, very directly. And this is not something that's unknown. I mean, when America had its own civil war in the 1860s, the doctors knew that the only real way to save the patient was, in some cases, cutting off the limb where the bullet or the infection had occurred. The problem was they didn't have sterile technique in the 1860s, and so invariably, 100% of all patients that went to the doctor, to the physician's hospital or the medical hospital, the military hospitals, basically got an infection afterwards. And many of those succumbed, that is, died because of that. Now, when Fleming made this observation and wrote it up, you have to understand that there was a lull. The only previous situation of chemical therapeutics against bacterial infections was really around the turn of the century by Ehrlich, who developed an arsenic compound called 606, and it was used to treat syphilis. And of course, you had a certain distance between, and when I say by distance, you had a certain amount that you could apply to someone before they would get sick from the arsenic of the arsenic compound. But for the first time, they actually had a chemotherapeutic approach to syphilis. Now, what happened as World War II started ramping up, a variety of government and private companies began working on ramping up production of and extraction of and mass production of penicillin. Okay. Now, also, there was another noted drug, prototocil, which was a red dye. It was effective against streptococcal bacteria. Also, you had the discovery of streptomycin, which was really extracted from Streptomyces griseus, which is another microbial organism. What you're seeing is this revolutionary turn of events in the way that modern medicine was occurring. from before World War II till afterwards. And it was dramatic. Instead of a lot more pain and suffering, people were able to walk away after a serious bacterial infection or serious wound, etc. Now, antimicrobial drugs are described by the pathogens they target and their mechanisms of action. There are two things you have to be aware of. There is the broad spectrum drugs, which are effective against a spectrum of organisms. In other words, they're effective against gram-negative and gram-positive bacterial cells. Narrow spectrum drugs, they target a limited range of bacteria. They're preferred in treatment to avoid disruption of the normal microbiota. What you'll find is narrow spectrum, a lot of times the doctor has to know what he's dealing with before he would issue a narrow spectrum drug. Now, there is a concept called empirical drug therapy treatment, and that's based on clinical presentation in the absence of definitive or complete medical data. So we look at these, basically these clinical signs. Based on this, in the absence of a much more broader studies, someone may use a drug. There is another set of terms that you need to be aware of, and that is bacteriostatic versus bactericidal. Bacteriostatic are drugs that prevent the growth of the bacteria. So yes, the bacteria are still alive. What is hoped is that they've been kind of put in almost a stasis. What I mean by that is they cannot reproduce. No binary fission is going to occur. And hopefully, the patient's own immune system will be able to recoup the response to the bacteria that are presently there. Now, bactericidal, these are drugs that actively kill the bacteria. On the left, you see a recreation of the mold-contaminated culture plate, and this led to Alexander Fleming's discovery. If you notice, there's a lot of little colonies here, but around the mold itself, no. And so there was diffusion of whatever products being produced by the mold into the medium and made it either basically killing the bacteria or inhibiting the bacterial growth. This is a colony of Streptomyces griseus, and it's basically a soil bacteria that makes streptomycin. Now, antibiotic drugs can be natural, synthetic, or semi-synthetic. Let's go through each definition here. Natural is extracted from the bacteria or the molds themselves. The original penicillins were extracted from molds that were in various types of culture. Synthetic, they're entirely manufactured by chemical processes, so they usually avoid antibiotic resistance mechanisms. So if you remember, or if you had any organic chemistry, it's basically when we take this molecule, we add this methyl group here and this hydroxyl group here, and this group we might put an ethyl group here. And what happens is that basically it is a process of synthetic chemistry that ends up formulating the material and producing a large batches of it. Now, you can do another process where basically you modify the antimicrobial drug. And this can be done to enhance its half-life, to avoid antibiotic resistance, to making the drug more water soluble, for example, much more soluble in plasma, much more easier to uptake from the gastrointestinal system to the blood. For example, adding a fluorine atom to the molecule, such as with fluoroquinolines, or adding groups to the penicillin, which we'll see in a diagram. Fluorine tends to have a much stronger bond, so it's much more resistant to enzymatic action, let's say by pathogens like bacteria, etc., to remove it. And so it tends to have a longer half-life as a result. Then we have to talk about the different types of generation of drugs. First generation drugs, drugs that have undergone first round of chemical modification. Second generation, drugs that have undergone second round of chemical modification. Usually this is to expand upon capabilities over the predecessors. So in some cases, to help you understand that, it's as if you take a narrow spectrum drug and by enhancing the structure, you now make it a broad spectrum drug for that example. And there are 5th generation drugs out there now that have been modified in small or larger means by adding on groups. And as a result, you now have it much more resistant to the drug-resistant enzymes that some bacteria make, or you may allow it so that it is quite stable to pass in through the digestive system and eventually get into the circulatory system. Now, if you take a look here, here's a chart. As I was mentioning earlier, penicillin G, it was the natural product, 1943. And then what happened was we started to find ways to add on. And this is where you get ampicillin, which was a semisynthetic molecule that came out 1961. And all they did was they added an amino group. Then you get to the semisynthetic next generation, which is the amoxicillin, excuse me. And that came 11 years later, where they added not only, you have, of course, your amino group, but now they put a hydroxyl group there. So there you go from having a natural, basically extracted from the mold, antibiotic, going through 2 rounds of this, and they're still around here today, these drugs. Now, a number of factors impact antimicrobial drug development. First, of course, is drug safety. Some of these factors of drug safety basically limit the use in the body. Sometimes the limits make the drug useful and non-toxic only for topical use and for parenteral applications in other cases. Let me give it to you as a simple way to think about this. You go out at home, open up your first aid kit or something else like that. Maybe you've got a medicine cabinet and you see these topical antibiotics. The reason that they are there, how come it says bactericin and polymyxin B and so on and so forth? And what's this neomycin? These are mostly drugs that are put in as creams or topical uses because you cannot use them intravenously. You cannot use them orally. Okay. Then we have the issue of selective toxicity. This is the core principle that drugs must inhibit or kill the target microbe without damaging host cells. This is something that we've learned a long time moving forward. There were some antibiotics that in the beginning they were great, but then they started noticing that they would cause deafness by destroying some of the hair cells in the inner ear, or they might cause damage to the kidneys or the liver. This is where you get into the concept of side effects and total risk-benefit analysis. It's sort of a consideration of, well, what's the adverse effects versus the advantage to kill the pathogen? Let me explain this to you from just going back to salvaricin, which was that arsenic compound I told you about earlier. At 1900, they had no way to treat syphilis. Some of the elite would still turn to a mercury injection, and that had severe toxic effects and didn't really kill off the syphilis. What happened, though, was, well, salvaricin, compound 606, does have some toxic effects, but it was the best that was available at the time to treat individuals who had gotten syphilis, because there was no other treatment for syphilis, and what meant is that you had eventually going down neurodegeneration and sterility and all these others. I shouldn't say as much sterility as it is, that you would have serious problems. So once someone got syphilis, they really could not marry, and that's where you get into the tests that they have, blood tests for marriage. It wasn't just testing for HIV. Before even the era of HIV, it was testing for syphilis. And what happens also is that syphilis, if the individual did not know it, could also be passed on in utero to the developing fetus, and you would have adverse effects. You could have stillbirth, deafness, heart defects, all sorts of very sad effects. So when salvaricin came out, there was a mixed review, but it was somewhat favorable because beats nothing. And nowadays, what we talk about is the therapeutic index, otherwise known as the therapeutic ratio, which is basically the maximum safe dose divided by the minimum effective dose. Now, you know what the minimum effective dose is. That is the minimum amount that will actually serve to fight off the microbe, kill off the microbe, etc. The maximum safe dose is how much is the maximum that a person can take without any adverse effects or without serious adverse effects. And I mentioned this a while back to some students, and I'll mention it again. This is why we end up having drug trials that go through phase 1, phase 2, phase 3. Phase 1 really looks at the safety parameters. And so what happens is you have a small group that's given the drug at 3 different doses to see if there is any maximum safe dose compared to the minimum effective dose, if there's a minimum dose that could be used, if there are any adverse reactions, etc. Now, always a high therapeutic index is favored over a narrow therapeutic index. What I mean by that is, if you've got, let's say, I'm just going to pull numbers off the top of my head. Let's say your maximum safe dose would be 1 gram per kilogram body weight versus the minimum effective dose is 0.001 micrograms per kilogram. Well, that's a really whopping difference. But if, let's say, your dosage, the minimum effective dose was, let's say, half a gram per kilogram of body weight, and the maximum safe dose is 1.25 grams, you have a narrower range. And what happens, you have to keep in mind, is that this is all based on large group studies, etc. You're going to have individuals who will have a faster metabolism, a slower metabolism. Others that are going to have other physiological factors that may lead to adverse effects. Now we get into therapeutic drug monitoring, TDM. This is measuring the drug parameters of the patient, and that is basically the blood concentration during the treatment, while monitoring for adverse effects. So you might be using the drug and monitoring, body temperature, heart rate, respiration rate, kidney function, looking for signs of any liver damage, etc. Toxicity considerations. You remember what the organs of the body are involved with filtration and detoxification. Here we are. Liver, kidneys. If those are affected, you have a really serious time. The kidneys don't have as much of a bounce back as the liver does. You can have a certain amount of damage to the liver and it will regenerate. Kidneys, not as much, but you can reverse some effects by just stopping the dosage. This is where we get into the terms. Nephrotoxic, this is toxic to the kidney. This includes a very long list, but at the top of the list are the aminoglycosides, okay? Then we've got hepatotoxic, and this is toxic to the liver. The liver has a greater capacity to rebound or regenerate, as I mentioned before. And then we have this unusual acronym DILI, drug-induced liver injury. This is damage to the liver introduced by drugs, especially antibiotics. What you have to consider also is the patient on other medications at the time that they're given the antibiotics? Because you may already be stressing the liver, and the antibiotics may actually push the liver into further damage. Also, and I'm going to mention it here now, but I'll mention it probably later on too, this is why today you listen to doctors and you listen to the medical interview, they get the case history. They will ask very directly, has this patient been on anything outside in the sense of herbs or special diets, etc., because some herbal compounds will actually interfere with or have a certain level of hepatotoxicity, and you can have a synergistic effect. Some of it coming from the antibiotics, some coming from some of the herbs the individual had. Herbs, you have to understand, yes, sometimes they have some benefit, but herbs are not a pure substance. Any herb that you have, and I remember this from one USDA ethnobotanist who basically went down and looked at a lot of the different herb compounds and herbal makeups of a variety of cultures. He's an ethnobotanist, means he goes to different cultures and looks at what they have and what they use and why, and then maybe trying to isolate what are the active compounds. There are some herbal compounds that have an effect of raising blood sugar, raising blood pressure. Also, they can be abortifacient. And that's not what you want to have certain individuals get. Let me move on. When we talk about administrative route, the procedure by which the drug is administered to the patient, it can be oral, it can be in a pill, a solution. It can be parenteral administration, which is infusion or injection. When we talk about injections, the drug could be administered via injection or infusion intravenously, intramuscularly, or subcutaneously, and that depends on how fast the substance must get into the circulation, to counteract the damage that's being caused by the microbes. Drug stability and elimination. When we talk about half-life, this is the time it takes half a drug dose to be eliminated or deactivated by the body. Now keep in mind the two terms, eliminated, deactivated. Elimination, well, that's easy. We just talk about it. It gets excreted out via urine or feces, or in some cases both. But when you talk about deactivation, this can be coming from a variety of enzymes and other biological effects. For example, remember that once you consume something orally, it goes into the stomach. There it's exposed to a proteolytic enzyme, pepsin, but it's also exposed to hydrochloric acid. And then after that solution, a chyme as they would call it, goes from the stomach into the duodenum, now you've got a variety of other enzymes that will act on it to perhaps break down some of the compounds. They may also, because of the bicarbonate ions released from the pancreas, etc., have other effects on them. Just trying to give you some idea about some of the points here. Now, drug interactions and contraindications. It is extremely important to understand that some antibiotics can blunt the effectiveness of some drugs, can work in competition with other drugs, and even in some cases, you will have a synergistic or a magnified effect. Not necessarily to fight off the, let's say, fighting off the microbe, but it could be having an effect that might raise blood pressure, increase urinary output, or damage the kidneys or the livers, as I mentioned before. Now, one of the known situations is antibiotics can blunt the effectiveness of oral contraceptives. I have a friend years ago and came back and said, I'm going to be a grandmother again. I said, well, congratulations. And she said, yeah, and it's all because my daughter was on antibiotics because the antibiotics were blunting the effectiveness of her oral contraceptives, and she got a little grandson as a result. But what about grapefruit juice? Well, grapefruit juice is something that came out in the later 90s and into the 2000s. It turns out that grapefruit juice contains furanocoumarins. Now, basically, it's components that make up part of the grapefruit juice, but it blocks an enzyme called cytochrome P450 3A4, which is a mouthful, but just keep in mind this. This is an enzyme that normally would break down many drugs in the intestine and the liver. For example, clarithromycin. Okay, now, What this happens to do is it lengthens the half-life of that antibiotic much longer. And it also may account for why you have sometimes incredibly long plateaus or peaks in the drug levels in the blood. So if you think about it, well, the doctor says, well, it'll take one a day, but they're not being effectively broken down and not effectively being excreted. So what happens is after a few days, you start having a peak level of the drug drifting further and further, higher and higher, which may trigger adverse effects. Okay, I hope that helps. Now, antibacterial drugs may be grouped by their cellular targets, and I refer you over to figure 15.4. We'll get into that in a second. Many of the antimicrobial drugs disrupt bacterial cell wall production. You know that bacterial cell wall stability is essential to maintain the integrity of the bacteria cell, particularly where you have osmotic forces, water concentrations inside versus outside of the cell, etc. So to maintain the structure and the shape of the cell, etc., you have to have a very strong cell wall. Now, part of this is produced through a process called transpeptidation. It's a process where the sugar molecules are going to cross-link with peptides in the cell wall. Now, antimicrobials that target cell walls include penicillin, cephalosporins, carbamazepines, monobactams. All of these have the beta-lactam ring. It seems that the beta-lactam ring is a four-sided structure. It is also the target of antibiotic-resistant enzymes. This is where you get into beta-lactamase. It's an enzyme found in bacteria with antibiotic resistance. And beta-lactamase inhibitors have been developed, clavulonate and sulfactam and tazobactam. Now, here's the key. This is what goes on. You probably have heard of the drug Augmentin. So what this is, a mix of amoxicillin and clavulonate, because amoxicillin would be normally disabled by the beta-lactamase enzyme. No problem. Clavulonate blocks the beta-lactamase enzyme so amoxicillin can do its damage necessary to the bacteria cell walls, and boom, the cells break up, they lyse. Here you can see in this diagram here, everywhere you see in the green, that is the beta-lactam ring. And you can see the monobactams, you can see the structures to the left, structures to the right, extensions off of the nitrogen in that beta-lactam ring. And this is the fascinating part about all of this, connecting the structure. You have all the different types of penicillins, carbapenems, the cephalosporins, etc. What that key means is this: the beta-lactam ring is an essential structural configuration that is necessary for the proper function of these cell wall-based or targeted antibiotics. But the enzyme beta-lactamase can destroy all of this and render them completely useless. Here you see the sugar chains and the peptidoglycan and the protein crosslinks. What happens with the transpeptidase enzyme that's supposed to make these linkages? Penicillin blocks it. So what you see on the bottom here is cells blowing apart, breaking up, because they lack the crosslinks necessary for the structural integrity of that cell wall. Here you have a normal cell walls. You see them. They are bacilli shaped. These are the cells that are falling apart. They're lysing, bursting. Okay? Now, let's talk about penicillin. All of these in the penicillin family end with -cillin, ampicillin, penicillin, etc. We start off with penicillin G and then we move forward. Penicillin V, orally only. Ampicillin is part of these many broad spectrum applications. Now we have to get used to some of these acronyms. MRSA means methicillin resistant Staphylococcus aureus. ORSRA is oxacillin-resistant Staphylococcus aureus. And then MDR means multi-drug resistance. Now, to help you to understand this, a lot of times with MDR, they don't just have a particular enzyme. They may have several other factors that make it extremely difficult to try to treat. They've either developed enzymes and other forms of defense against antibiotic resistance. We'll talk more about that in a bit. But MDR, and then you have XDR or XMDR, and that is extended multi-drug resistance. There is, for example, one type of gonorrhea that may have gone to the point of almost being completely resistant to all present antibiotic or antimicrobial drugs. This started off from the super gonorrhea, which had MDR resistance, and it was found in Korea and in some of the places nearby military bases, Japan, Guam, etc. Why I bring these up is because in other places of the world, there are other sites that are having also MDR resistance. Russian prisons have a tremendous amount of tuberculosis, and many of these tuberculosis strains are XDR, meaning virtually most of the drugs that are used to treat TB are resistant because the tuberculosis organism has many, many defenses against a variety of antibiotics. And so the fear is that in these prisons that have been allowing this to occur, individuals are dying in there. And it's also scary if you would turn around and say, well, do we let them out? If we let them out, then we spread this extremely drug-resistant tuberculosis to the general population. I'm just reporting this. I'm not making a judgment. I just wanted everybody to understand that. Cephalosporins, they're broad spectrum. They constitute the largest collection of beta-lactam drugs. Usually when you're talking about something in the cephalosporin family, you have a prefix of either CEF, Ceph, or CEPH. A fifth generation ceftaroline, otherwise known as teflaro, is used against MRSA and ORSRA. Also, carbapenems, same target. The problem that they have is there's a risk of seizures for kidney patients using this drug. It has been found to be effective against MDR bacteria. Monobactams has only one ring. Only one of the monobactam family is in use, and that's azotrinom. And it's useful against gram-negative bacteria. Now, if you can see here in this diagram, we have cell wall synthesis, that's a target. Plasma membrane, that's a target. You have polypeptide drugs, which we'll talk about afterwards. In the nucleic acid area, you have quinolones, and rifamycins. For protein synthesis, see that? That's the ribosome. If you remember, we talked about ribosomes much earlier in an earlier lecture. That is a key target for many different types of antibiotics because they will work on this. And as they do this, usually though we start shifting from a bactericidal, meaning that we kill the bacteria, to a bacteriostatic, which means we stop growth. Finally, folic acid synthesis, and this is with sulfur drugs and trimethoprim, and they block the enzymes that are part of the folic acid synthesis chain. Moving forward now, before you lose any sleep, I would like you to look over these, but do not memorize. Everybody got that? What this means is focus on the presentation, but don't lose sleep on each and every one of the little drug names, okay? Just get a lot of the basics and follow with what is on the PowerPoint slides, okay? It is important to get an idea about what we mean by when we say broad spectrum, narrow spectrum, bactericidal, bacteriostatic, and what are the targets. Here's an interesting thing, penicillin allergy rash. We talked about some of this as hypersensitivities in a previous lecture. And so you can see this kind of rash, this modeling that occurs after the person has started to use penicillin. Now, Glycopeptides, these are non-beta-lactam. So they lack the beta-lactam ring, but these are antimicrobials that target bacterial cell walls. An example, vancomycin. Now, you do not have beta-lactam rings, so you're not susceptible to beta-lactamase. Great. Some of these drugs, though, are narrow spectrum. They're useful against some antibiotic-resistant strains and some gram-positive strains, and some MRSA with newer generations of the glycopeptides. But there is a particular adverse effect that will show up. It's called red band syndrome, and that's a red flush of the skin with an itchy rash. This has been mistaken for a drug allergy. It's really a side effect of certain glycopeptides. So here we have the molecular structure of vancomycin here, and you can see that central ring there. And here we have an adverse reaction, red band syndrome, to vancomycin. Okay. Now, there are other miscellaneous non-beta-lactam antimicrobials that do target bacterial cell walls. Bactitracin, commonly topically applied as neosporin. It targets the cell walls. It is a narrow spectrum against gram-positive bacteria. Isoniazid, which is used to treat tuberculosis because it interferes with mycolic acid construction in the cell walls, which you see with Mycobacterium tuberculosis. Okay. Here's our bactitracin. Look at the huge molecule it is. But if you take a look at something like isoniazid, look at the simplicity of that molecule, just a phenol ring with a amine group here, and then a short chain off of this. This, you can see, is a first aid antibiotic ointment that basically you would use for minor cuts, scrapes, burns, all topical application. This you use for anti-tuberculosis and can be taken orally as tablets. Quinolones and rapamycins target nucleic acids. Now, what quinolones do is they target the DNA replication enzyme. So DNA gyrase, DNA topoisomerase, as they have to separate out the two strands of DNA to begin the semi-conservative replication. The interesting thing is that one of the most commonly modified applications is adding a fluorine. Hence, we have the fluoroquinolines. And in that family includes ciprofloxacin, Cipro, and levofloxacin, okay? And I believe they call it Levaquin for its name. Usually these are targeting Mycobacterium, but they have been useful in other situations. Most commonly and widely remembered was the distribution of Cipro after the Amera anthrax situation that occurred in 2001, because although some people were mistaken thinking they just take a couple of Cipro and they're fine, you really have to be on 60 days to prevent any exposure to lead into an infection for anthrax spores. Now, rifamycin originally was isolated from bacteria. It's now produced as a synthetic and a semi-synthetic version. It inhibits the transcription by binding to RNA polymerase. Different target, but similar, nucleic acid. It is a broad spectrum antibiotic against gram-positive and some gram-negative. Here we have the molecular structure of vancomycin, as you can see, but also we have ciprofloxacin. And if you notice in that green there is the qualification for being a fluoroquinoline. Without it, if this was just maybe either carbon or hydrogen or anything, it would be just a plain old quinolone. Okay? Now, moving forward. Antifoliate drugs target folic acid production. One of the things you have to understand is that folic acid is necessary for DNA replication and DNA synthesis, and the foliates are extremely important. This is where we get into the onset of sulfa drugs. They originally were discovered in the 1930s. The first synthetic antimicrobials were used to combat bacterial infections. They act as competitive inhibitors of folic acid production, and I'll show you that in a minute. The bacteriostatic, though, is the end result. They have a broad spectrum of action, but they are bacteriostatic as opposed to bactericidal. Para-aminobenzoic acid, PABA, is necessary for folic acid synthesis. Now, sulfametoxazole is similar in structure to PABA. So what would happen is this sulfa drug will fit into the same active site of the enzyme that's involved with folic acid synthesis and basically shut down the production line. Now, there's another drug called trimethoprim, which is, although not a sulfa drug, it is also an inhibitor of another folic acid synthesis step a little bit further down. And together, these two drugs, trimethoprim and sulfametoxazole, you have a synergism, a combined effect that's greater than the sum of each type used alone, which pretty much puts the cell into sort of stopping all the mechanism. Here is the example of the para-aminobenzoic acid. As you can see down here, we've got this little grouping here, which would be a carboxylic acid grouping. Okay, so here's the hydroxyl group, here's the ketone group. Notice you've got the similar thing, but also what happens is instead of carbon, you're having a sulfur here. So this is the sulfanilamide that you see. They added an amino group, and that would fit right into the enzyme active site and basically shut it down. Now, there is another compound, which we have probably never really seen, but it is extremely important, particularly for burn patients. The sulfa drugs have been used to help individuals who have had very severe burns. Here you have the doctors, and I believe that's a nurse there, medical assistant, and they're basically going to cover up the burn areas with sulfur sulfdiazine. Now, sulfdiazine is a type of sulfur drug, but they add silver. And the presence of the silver also has an antimicrobial effect. This is all done where you have very severe burns. We're not talking first degree nor even necessarily second degree. We're talking third degree, and it's applied on top to prevent Pseudomonas aeruginosa and other bacteria which would normally colonize dead or debrided areas due to third degree burns. Now, some of the drugs target prokaryotic ribosomes. Remember that the prokaryotic ribosomes are different in structure and configuration, in size, than eukaryotic. Because of that, you can give a drug and not necessarily interfere with translation of eukaryotic cells, as you will be able to affect and shut down translation, the making of the polypeptides at the ribosome site for bacteria. Macrolides will target the 50S subunit of the bacterial ribosome. They block protein synthesis. These are of the class that you probably see commonly: erythromycin, azithromycin. Those are the Z-PACs you've seen. Clarithromycin. They're usually a broad spectrum antibiotic. LINCOAMIDES bind to the 50S subunit. They block protein synthesis. They're a broad spectrum. The problem is use of them increases the risk of Clostridium difficile. Henceforth, I'm going to refer to that as C. diff. And this is a really, really messy and difficult problem for healthcare institutions because once it starts breaking out, it can go very quickly and make a lot of patients sick and make an absolute mess. This is C. diff, pseudomembranous colitis is usually something that takes a very concerted effort. So lincoamides are not used in what would be called healthcare institutions as much. This could be nursing homes, it could be a hospital, etc. basically just to reduce the risk of C. diff. PHENICOLs, these bind to the 50S subunit. The main one is chloramphenicol. It's effective against many gram-positive and gram-negative bacteria, but has a very narrow therapeutic index. Why? Because it can lead to aplastic anemia, so the patient usually has to be monitored in hopes of not having that develop. Tetracyclines, and these include, of course, tetracycline, demiocycline, and demiclonone-cycline, sorry about that, as well as doxycycline, doxy as people would call it. This is different. It binds to the 30S subunit. It's broad spectrum, but again, it also increases the risk of C. diff. Tetracycline should not be given to children under the age of 8 because they tend to start having this cause what is sort of a side effect. It's teeth staining. And so once you get that teeth staining there, it doesn't go away. Aminoglycosides bind to the 30S subunit. These type of drugs block protein synthesis, but again, another increased risk of C. diff. Aminoglycosides have a narrow spectrum. They're given intravenously. They have a poor intestinal absorption. They have a short half-life, 2 to 3 hours. Rarely used alone. Usually what happens is you give this as a 1-2 punch or 1-2-3 punch with other antibiotics at the same time. Commonly used topically is neomycin. Non-topical use with, of course, includes streptomycin, amikacin, tobromycin, and gentamicin. Okay. So you can see in this chart here, obviously we're going to have the larger subunit, that's the 50S ribosome, the smaller subunit, that's the 30S. Macrolides, lincosamines, and chloramphenicol will go after 50S. The 30S is tetracyclines and aminoglycosides. Here's an example of the teeth staining. You see that dark band there? That may have been a time in which, as the tooth was growing and developing, the exposure of tetracycline showed up. Okay? And let's move on. Now, there are certain polypeptide drugs that target membrane structures. Gram-negative outer membrane will act as a selective barrier to some antibiotics. Polypeptides will be able to overcome this barrier. Both have a narrow therapeutic index. Therefore, they're mostly used topically. Let me start off with the first one, polymyxin B. This is narrow spectrum and targets the outer membrane of gram-negative cells. It's used fantastically for topical use for cuts, scrapes. It's usually together as a triple antibiotic ointment with several other compounds like bactericidin. Colistin. Oh, that is the last resort option. Okay, let me explain to you. Also known as polymyxin E. This drug is not frequently used in typical care because of its side effects. Okay, let me explain to you that if you hear of a multi-drug resistance and they find that it's resistant to colistin, all bets are off. I'm not going to say that the end of the world comes, but I am saying that you really have a serious crisis there because you've got literally an organism that has no drug is really going to be effective against that organism. And so it's a pretty scary situation if this gets spread to other people. How do you treat a patient who has this particular extra drug resistance that includes the last bastion, colistin? Now, I have basically said everything I can see there. Truly untreatable infections. Well, let me move on. You can see here, here's the polymyxin. It begins to attach to a lipopolysaccharide, and it begins to interact with the phospholipids and disrupt the outer membrane. Eventually, it will transverse the peptidoglycan layer and disrupt the plasma membrane. Now, we're moving out of antimicrobials that are focusing on bacterial infections. Here is a tip to help you. The more unique the genetic makeup of the organism as compared to, let's say, humans, the better chance you're going to find some drugs that will work and target the elimination of that organism as an infectious agent. The more similar you have, the less likely or the much more difficult it will be to develop antimicrobial compounds against that particular organism. So to help you to think about it this way, bacteria were easy to develop in some relative means for antibiotics because bacteria are prokaryotic. They have a different size of their ribosomes. They have different compositions that hold together their cells, cell wall, etc. The closer you get toward humans, such as multicellular, eukaryotic organisms, the harder it is to find a compound that will uniquely affect the pathogen, but not hurt the host. That's where I'm going with. First, though, let's take a step aside and get into viruses. Antiviral drugs target specific points in the viral replication, whether it's attachment, penetration, uncoating, replication and viral assembly, release. There is a third, there's sort of like a sixth class, the last one, interferons. And what they do is stimulate the immune responses against the viruses. They can also act as flags to neighboring cells that are infected, that are not infected with the virus yet, but the cell that is infected with the virus can sort of like notify its neighbors, I'm infected, you don't want to associate with me, or you want to set up your defenses. Drugs are effective against viral infections, but latent viral infections are much more difficult to treat. That being said, you can use some, but it's almost not as efficacious, and it's been something that hasn't always been an easy route. Case in point, although we have individuals that are now living 20 plus years with HIV infection, and their viral titers are 0, there have been studies where they tried to see, well, if they've had a year without the viral titers present, let's remove them from the medication they're on. And so they get off the medication and the viral titers take off. There must be some places where the virus is hiding, that it basically stays in stasis and will not become active again until the antiviral medication is eliminated. Same thing would be for the chickenpox virus, where you have a situation where it may be 50, 60 years later and the individual starts getting shingles. And there are other situations. But I want you to just review the diagram briefly here. I don't expect you to memorize all of this because in part, a lot of these drugs are for unique viral infections, such as hepatitis B virus, hepatitis C virus, rabies, HIV. You will see HIV also with replication and assembly blockers, as well as protease inhibitors. Release is blocked by 2 drugs that have been applied for influenza, zanamivir and oseltamivir. You also have rimandidine and amandidine. Now, there's 2 points here. This is uncoating blocked by and from these drugs. How come we don't get these? How come they're not over the counter yet? How come this? How come that? Basically, antiviral drugs are not released to the public willy-nilly because doctors are afraid that you're going to have the same problem of antiviral compounds eventually leading to viruses that become resistant to these antiviral compounds. Is it possible? Yes. Is it inevitable? I don't know. But I do know this. If you freely gave out these as much as people would freely get antibiotics, we would eventually start seeing some of these effects. And when we get into resistance, let me remind you of one little interesting fact. Penicillin started taking off in its use about 1943, 1942. Okay? Fine. By 1946, the first case of penicillin resistance was reported. Didn't take a long time. It just took more cases building up for the medical community to really take notice. Let's talk about antifungal drugs here. Remember the structure of fungus? It's eukaryotic. It has hyphae. It will release exoenzymes to basically break down the tissue to absorb the nutrients from the breakdown products. But antifungal drugs often target the cell wall and membrane structures. Now, those that target fungal plasma membranes, these include the azoles and aluminine, which inhibit enzymes that synthesize ergosterols, which are present in the plasma membranes of these fungal cells. What you end up having is improperly formed cell membranes as a result of these drugs that lead to fungal cell lysis. Now, azoles, such as fluconazole and ketoconazole, these are broad spectrum agents. They're used for such things as athlete's foot, ringworm, yeast infections. The aluminines are terbinafine and naphtidafine. Now, the polyenes, such as amphotericin B and nystatin, interact with ergosterols, causing cell membranes to become leaky. Now, amphotericin B has an issue with toxicity. Therefore, its use beyond topical, such as for intravenous, etc., is reserved only for life-threatening systemic fungal infections. Nystatin itself is too toxic and therefore is only used topically. It works by inhibiting the fungal cell wall synthesis. Now, echinocandin drugs, such as caspofungin acetate, these inhibit cell wall synthesis by targeting the enzyme making beta-glucan A. Now, this is a component, beta-glucan, and this is a component of the fungal cell walls. It's used mostly, though, for immunocompromised patients with systemic fungal infections. Also, when we talk about antifungal drugs, we have those that target inhibiting the nucleic acid synthesis, such as flucytosine. This targets DNA replication and blocks transcription. You see, upon entry into the fungal cell, this is converted to the nucleic acid analog that blocks DNA and RNA synthesis. Here you see naphtidafine, and you can see how it's long. It has several of these benzyl rings, and the gentleman is applying this as a cream for athlete's foot. Now, here's where we get into even more of a challenge, because now you're dealing with something that is closer. I mean, yes, there's a difference between a parasitic worm and a protozoan and myself, but there are things that are similar. That is, we're multicellular and we are eukaryotic. And so the ribosomes are similar. We don't necessarily have cell walls. We have cell membranes. And so it gets more interesting how we're able to target the intracellular components of the protozoans and the helminths. Okay? One of the key ones that gets everyone's interest first and foremost is drugs that affect malaria. So we're going to talk about antiprotozoal drugs, and we divide them into antimalarial drugs and non-antimalarial drugs. Anti-malarial drugs are in 6 classes: aminoquinolines, aryl amino alcohols, artemisins, respiratory chain inhibitors, antifoliates, certain antibacterial drugs such as doxycycline and clindamycin. Well, what about chloroquine? This is an aminoquinoline. It's most widely used. Unfortunately, after decades of its use, malaria resistance has become widespread. It used to be a time where you would go to, let's say you were going to go to a part of the world where malaria was quite present. You could take chloroquine tablets when you got there, maybe a little bit before, and then until you left there a little bit afterwards, and pretty much protected yourself from getting malaria. Unfortunately now, wide swaths of areas such as Asia and South America and Africa have malaria-resistant protozoal parasites available. So they change over to some other drug, such as quinoline. But where did chloroquine actually come from? Chloroquine was actually found to be a modified compound from quinine. Yep. The earliest compounds given were quinine, which is an extract of a cinchona tree. It's from their bark. Now, to help you understand this, it actually has a cultural effect. In the time of the 1800s, when British troops were occupying India, malaria could wipe out all of those troops. So what they started doing was making sure that all the troops would have quinine. But quinine is exceedingly bitter. And so what they did was they started to give a sweet tasting liquor made from yew berries, okay, and it was called gin. The quinine was in a watery solution called tonic. Yes, that's where you get gin and tonic. And it was basically distributed to all the troops there so they would not get malaria. Okay. Now, methoclin is another quinine derivative. Unfortunately, they're reporting resistance there too. It's used to kill the parasite only in the red blood cells. Now, you have to understand, if you go through some of the parasitology, you'll find that malaria has several stages, and one of the stages will go into the liver cells, and then another one will go into the red blood cells. And it is infecting the red blood cells that it will be picked up by the mosquito again and pass through its life cycle in the mosquito and then come out through the mosquito's salivary glands and the next bite for blood the next time around. Now, there was an intriguing piece of information coming from an extract from Chinese herb, sweet wormwood. This was called artemisinin. And the actual researcher who did the work, she was looking at, do any of these active compounds are really coming and really having an effect based on ancient Chinese medicine? And she found that the Chinese herb, sweet wormwood, had one. It was called artemisinin. It provided good effects. Sadly, also resistance is being reported now, particularly in areas of Southeast Asia, etc. Now, artemisinin-based combination therapies, ACT, use artemisinin-based drugs in combination with non-artemisinin drugs to treat simple malaria cases. That way, when you have sort of this drug cocktail, you prevent resistance. It's one thing for an organism to have a mutation or develop a mutation of resistance against one drug, but not several at the same time. And so usually what happens is you're able to get rid of them. And what I mean by simple malaria cases, let me just help you that for human malaria, there are 4 different parasitic organisms in the Plasmodium group that will lead to malaria. Usually individuals that have all four will die very quickly. But if it's a simple malaria case where they can see and isolate and say it's, Plasmodium vivax or Plasmodium molvax, etc., they can move forward with the ACT. Now let's move from here to non-malarial antiprotozoal drugs. The best known, Flagyl, otherwise known as metronidazole. And this is basically nitroimidazole, which is used to treat the protozoal parasites. Now, which ones are we talking about? Giardia, toxoplasmosis, trichomoniasis. It's also used to treat mild to moderate C. diff cases, which is interesting. Remember, C. diff is a bacterial infection. Also, they found that using TMP-SMX, which is the trimethoprim sulfametoxazole, will block the foliate production in protozoans. Nitrazole anoxazole blocks the aerobic energy metabolism in the protozoa. Here is the metronidazole, otherwise known as Flagyl. This is the package, and of course you can see the molecular structure here. Now we move from antiprotozoals to antihelminthic drugs. The most common are albendazole and mebendazole. The broad spectrum, they inhibit the glucose uptake by worms by targeting their microtubules. Praziquantel is used for fluke and tapeworm infections. And what that does is it paralyzes the worms. Therefore, the paralyzed worms are expelled in the feces. Now, we move from here to assessing sensitivity to antimicrobial drugs. Basically, what are the techniques done here? What you have is you really have to assess the bacterium's susceptibility to the antimicrobial drugs. It's absolutely essential for proper treatment, and you really have to understand, you're going to need a lot or a little. Is it going to last a long time or not? How long will it take to have effects on the infection or the microbe? Tests are necessary to identify susceptibility tests. These will determine if the drug is effective and the amount required. Now, this brings us back to agar diffusion tests, which include the following tests. Drugs basically will diffuse through the medium to kill or stop the bacterial, that is the microbial growth. Usually what you have is Mueller-Hinton agar. That's the agar that you're going to grow the bacteria on. And then you're going to have the tests applied onto that agar. Okay, so agar is going to the antimicrobial compounds will diffuse through the agar, and you'll be able to get an idea about the effects. Because if you've got basically an entire lawn, or basically all the bacteria spread out onto that plate, living on that agar, this is where we get into the Kirby-Bauer test, which is otherwise known as the disc diffusion test. You have paper discs with drug incorporated into the paper. They create a zone of inhibition. Now, the zone of inhibition is a clear zone around the Kirby-Bauer discs, which demonstrate the bacterium's response to the tested drug. If you've got a big, wide zone of inhibition, that drug is really good and effective at killing off the bacteria. If you barely have anything, no real clear zone around that paper disc, then what you have there is an organism that is resistant, or by some intrinsic means is not responsive to the drug. Now, there was sort of a step in advance from the Kirby-Bauer discs to the E-test strips. Now, here's the point. What is the amount? What's the gradient that would be applied? Or what is the minimum amount that is necessary to kill the bacteria? So, E-test strips are basically strips that have a variable gradient applied of that particular antibiotic on the strip. They will help provide the data for minimum inhibitory concentration. That is the minimum concentration of the drug that inhibits the growth of the bacteria. Here we see the zone of inhibition, the Kirby-Bauer discs here, 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 here. They're usually all around here, and what they have is, of course, basically a control. It's basically a paper disc, somewhere here that you're not going to see any reaction. If you notice, you can see from the color in this disc that it has spread out, and it's actually caused some pigmentation or coloration of the gel, the agar. Now, how would you measure a zone of inhibition? You go from one edge to the other through the middle of the disc. Yeah, well, there's nothing in the disc. Well, there wouldn't be anything anyways. So that's the Kirby-Bauer method, but here's the neat thing. These are the E discs. Now, they will usually have a couple letters designating what particular drug they are, and they will start at 1.0 and they will move upwards. And if you notice here, just as this sort of teardrop appearance occurs, what you've got about mid-range right here, about 0.25, that would be 0.25 micrograms per milliliter is the minimum inhibitory concentration. So if you draw a line right there, you got a little bit of it still here, back here. You have, and of course it's spreading out along the entire agar as you get higher and higher concentrations. So you know that it's being effectively inhibiting growth. Now if you take a look over here, you've got a spotty effect, something, but not a lot. Over here and over in here, you've got huge teardrops, cleared zones of inhibition. And that is how you're able to get a much more detailed look at the effect and the concentration of the drug necessary to inhibit growth. Now, of course, you've also got broth dilution tests, and this is a key test to differentiate bactericidal versus bacteriostatic action. The antibiotic is diluted in a series of concentrations on a microassay plate with the culture medium and the bacteria in each well. Now, if you get cloudiness, that's the buildup of bacteria cells. That's referred to as turbidity. And in that well, that would determine bacteria growth. Now, you use a colorimetric approach that you can use to indicate cell growth or cell viability. This is where you're going to get minimum bactericidal concentration, MBC. It's a concentration that is the lowest amount that will yield 99.9% in cell count. The reduction in the cell count, I should say. So, as you see where this microplate is right here, and you have where they put these large circles, that is, the first row, of course, is no drug in the row. When you have a clear circle right here, that tells you this concentration of this particular compound is effective. No growth occurs here at all. No growth occurs here. And you can get an idea about an increasing drug concentration as you go from top to bottom. And if you go left to right, you've got 12 different drugs here being tested, okay? You have increasing drug concentration, 12 different drugs, and of course you're going to have your control. So the first row has no drug in it at all. So you can see what you would have if there was no drug positive. You would have turbidity, you would have this coloration there, so you have growth present. And then you start going down in the or up in the concentration, and you see eventually the cells decreasing in their activity and eventually no cell at all. And so that's where you have your minimum inhibitory concentration. Okay. Drug resistance and proper antimicrobial drug stewardship. This comes back to what we were talking about earlier. Doc, I don't feel well. You have the flu. Yeah, give me some antibiotics. But antibiotics won't stop the flu virus. Give them to me anyways. And that's where doctors have had to learn to say no. You have to also understand that some pathogens have intrinsic and or acquired antimicrobial resistance. And Antimicrobial resistance, this is a situation where the microbe is not affected by the drug therapy that it was intended to inhibit or eliminate the pathogen. In other words, it used to treat the pathogen and kill it, now it does not. We now have superbugs. These are organisms that are resistant to the antimicrobials, and this is going to be a serious challenge as we move through the 21st century. We have situations of superinfection. It's an infection by the superbugs that is very difficult to treat. If the standard drugs don't work, then what else do we try? And we have to keep in mind that the person's normal microbiota has already been disrupted by some of the earlier drugs we used. But now it's a race against time. Who wins, the patient or the microbes? If you look here, you have the beginning of superinfection development. In any microbial population, there may be some that are resistant to the administered drug. The susceptible cells will all die. You will also have members of the normal microbiota eliminated as well. The resistant ones will thrive. A, in the absence of the competition. B, because basically you have no other drugs to treat at the time, you're not aware of this, and this will lead to a superinfection. The resistant microbes may also share resistant tools through horizontal gene transfer. This is something we talked about before. When we talk about horizontal gene transfer, we've talked about conjugation and the other types. Go back to genetics of the microbes in that chapter. Let's talk about intrinsic resistance, built-in qualities of a microbe that help them to naturally resist antimicrobial drugs. Acquired resistance is the only other way, and that's resistance to antimicrobial drugs due to acquiring genes that convey antimicrobial resistance. Usually this is done through horizontal gene transfer. Now, if you forgot about that, intrinsic examples, cell wall composition, endospore formation, things like that. When you have endospores, they're not going to be affected. And the genetic information inside is not going to be affected by antimicrobials. But let's move over to the acquired way. We have mutations. We eventually develop some resistance. And what happens is, now that you have those genes that have resistance, you're going to basically share them. Not realistically. I'm just trying to basically bring out the point. How will these sharings occur, this horizontal gene transfer? Conjugation. Remember that pili there? Transformation. The cell dies and you have this bringing in of these plasmids. Or transduction, that may be through viruses such as phages, etc., that will transfer this information over. Whichever way you look at it, the bacteria that once had this, and by the way, this jumps species. What do I mean by that? Well, you used to only see this in Staphylococcus. Now you see it in Streptococcus. You see it in Escherichia as an E. coli. You start seeing it in other ones, Borrelia, etc. Why? Because you're having these horizontal gene transfers in one way or another. There are three main ways microbes evade antimicrobial drugs. One, target alterations, mutations that alter the target for the drug binding and effect. Just to help you, remember, what's one of the targets? Ribosome. What's another one of the targets? Certain enzymes that you can alter and change maybe one or two sites, and suddenly that drug doesn't bind there and stop the process of binary fission, of DNA replication, of transcription or translation. Okay? Another thing that you can keep in mind is that some, and we'll shift over now to protozoal. Some protozoans have been found to shift the genes on the surface. Now, this is how they can evade the immune system. But through some mutations, they can also evade effects of anti-parasitic, anti-protozoal drugs. Drug inactivation. cells will produce enzymes that act to biochemically alter or break down the drug before it has time to affect the microbe. Simple example, beta-lactamases, enzymes that add phosphate groups to or acetyl groups to drug molecules. That's another thing. Also, reducing the drug concentration inside the cell. Lower amounts of the drug below the minimum inhibitory concentration is an important resistance method. How do you get it out? Well, one, you can limit the drug entry. Drug entry requires porin to enter the bacterial cells. These are the porin molecules. And changes in the porin number or structure alter the drug transport into the cell. We can pump the drugs out of the cell based on efflux pumps. What are efflux pumps? These are plasma membrane pumps that actively pump drugs out of cells. There are many types of efflux pumps that exist. Some are actually found on cancer cells and pump out anti-cancer drugs. So what do we have? Altering the drug's target, okay? Inactivating the drug. Well, if we're going to do altering the drug target, here's an example. An enzyme might not be as accepting at its active site for the antimicrobial. Slight mutations and targets altered. Efflux pumps pump the drug out as soon as it starts showing up, reducing the permeability. Or drug inactivation. This is the enzymes that we were talking about earlier that will break down or chemically modify that antimicrobial compound. Now, this is where we get back into that entire situation. I said, patient comes in, doctor, I'm feeling terrible. Well, you have influenza. Okay, so give me some antibiotics. But antibiotics don't help with influenza. Don't argue with me, doc, just give it to me. Doctor has to have the capability to say no. Human behaviors can accelerate drug resistance emergence, patient noncompliance, antibiotic resistance. This is a failure of the patient to completely use up the drug therapy term. And what I mean by that is, oh, I started feeling better, so I stopped using the pills, but you were given a 10-day dosage. You only went through 5 days. Well, that's okay. I'm good. I'm feeling better now. What they don't know is that shorter exposure may not have completely killed off all of the bacteria, but has really opened the door to some bacteria surviving and now developing and taking over that niche that has been provided. And as their fitness or survival is enhanced, at the same time, they have some resistance in one format or another. So you're favoring drug resistance for these particular organisms. Let's move into another area, antimicrobial misuse in agriculture and clinical settings. Resistance is accelerated by the widespread use of antibiotics in cattle. Now, the reason that they use this is to prevent diseases as well as to enhance growth and weight. And since the 70s, there's been, or 60s actually, there's been this push by certain companies to include in cattle feed or as a cattle feed supplement certain antibiotics. Some of them are not for human use, but are close enough in structure that this leads to the formation of some bacteria eventually coming out, usually in cattle poop and other things, excrement, yes. And what happens is suddenly, you have strains that are resistant to a class of drugs, for example, the penicillins and other things like that. Healthcare settings are perfect incubators for antimicrobial resistance. In some cases, that's where we've had some of the microorganisms because of improper use of drugs or because of improper cleanliness or whatever. Something has been allowed to run rampant in the facility, and as it has, it has developed resistance to a particular drug. I might also throw out that you have situations where diseases will run through a particular facility And if those facilities are poorly taken care of, this can lead to some very, very serious cases of multi-drug resistance. At the other end of the situation, as I mentioned earlier, like with the Russian prisons, there is very little care given to these individuals that are in the prisons, and there is tremendous amount of crowding, and so therefore you have the perfect incubation facilities for particular types of diseases that are easily communicable, such as tuberculosis. Okay. Also, resistant microbes have to be watched. CDC compiles a list of antibiotic-resistant strains and the development of new strains. So CDC should be informed when there is a new appearance of a resistant strain. They keep this on record and pass out alerts when new strains or resistance occurs. Now, you see here on both sides agricultural practices that promote resistance, clinical practices that promote resistance. I'd like to bring up one other thing. I included some short videos in YouTube that that are listed in the Blackboard in the last couple of weeks, those when we get into the topic of antimicrobial drugs, where they talk about resistance and they talk about some of the challenges. One of the challenges is, in some places, it's easier to get antimicrobial drugs. As a matter of fact, I'll give you an example. The difference between the United States and India. In a lot of places, you can buy over the counter what in this country, the United States, you would normally have to purchase with a prescription. Which means that a lot of times when people are feeling bad, they don't have time or money to the doctor. They will go to the nearest pharmacist and pick up whatever antibiotic they feel is necessary to use. And so what happens is that's why you have these rocketing numbers of antibiotic resistance in countries where the antibiotics are much easier to obtain, use, and thereby used improperly lead to onsets of antibiotic resistance. Okay. So, sort of a summary. Combating drug resistance requires proper drug stewardship. Yep. It's necessary to bring these to the attention of healthcare workers and patients to be able to say no to people who want antibiotics, but they want them, but they don't need them. And this is a key point. And I gave you the information about India versus the United States access to antibiotics. They have differing prescription rules, etc. There is another thing I thought I'd bring up also, just as we're starting to wrap down or wind up this entire lecture here. Developing new drugs, the challenges and opportunities. By the time we get about 1970s, doctors and companies were starting to shift all their resources away from antibiotic resistance. Excuse me, antibiotic development. Now I'll explain why. By that time, a lot of people felt, well, we got all these antibiotics out there we won't need anymore. We can make more funds and get new attacks set up with developing new drugs for cancer and heart disease and respiratory diseases and neurological. And so what happened was the antibiotic development stalled. Somebody that said, oh, I want to go into antibiotic research and maybe start developing, they were actually discouraged. So by the time you get to about the mid-1980s, it's pretty clear that antibiotic resistance is going rampant. So that's when new research began to take off again, as a lot of these antibiotic resistant strains exploded. Some of this was targeted drug design or structural molecular approaches, where they would look at the target, which might be the active site of an essential enzyme for viral replication or bacterial metabolism, and try to design a drug just for that. And unfortunately, developing an antibiotic, just like developing a lot of drugs, is not something that's 1, 2, 3, all done, start making the pills. Rather, the best estimates have been this. On average, from the time that you've developed the patent for your drug, you haven't started human trials or anything yet, to the time that you can get it on the market, it will average anywhere from 7 to 10 years. The average cost, $628 million. And that is if the drug is successful. If it's not, all that money is gone. In other words, all those fancy drug trials and everything else, and all the data that comes from the animal studies and all the necessary searching for adverse effects and everything else, and the clinical trials that have to occur, all of that is very, very expensive. And in some cases, I was watching pharmaceutical firms and new companies coming online in the 80s and 90s. What they would do is they would, as the expression goes, hook themselves to a star. They would come up and say to the larger pharmaceutical firms, look, we don't have the money and the manpower to do the clinical trials and to do also the marketing once we get our product online. So we'll make you a deal. And a lot of the bigger firms that had allowed a lot of their antibiotic resistance drug development to atrophy said, sure, we got the money, we've got the marketing team, and we can help set up your clinical trials. And that sometimes was very fortunate, very helpful, led to the development of new drugs. But even as we see now, new drugs are still being needed. So we have come to a conclusion here. We may not get into respiratory system infections, but I just wanted to encourage you to review the visual summary on page 462, review the chapter overview on page 463, review the end of chapter questions 465, 466, and also don't forget your homework in Mastering Microbiology as well as your quiz in Mastering Microbiology. Peruse the videos in the Blackboard for the lecture content. And have a wonderful day.